Hello, and welcome to this Microbiology Bio 203 video lecture from Chapter 18 of your OpenStax book, Adaptive Specific Host Defenses. I'm Mr. Kennedy, and I'll be your guide as we briefly explore this topic together. Learning outcomes for Chapter 18 include being able to describe the adaptive immune system and its two-arm humoral and cellular immunity responses, to be able to explain the types of antibodies and their actions, and to be able to explain how vaccines work. We begin today with an antigen. An antigen is a surface marker of a cell, a substance that can activate adaptive immunity. Antigens contain multiple epitopes. Specificity and memory. Your adaptive immune response will develop in response to each specific antigen that it encounters in a very specific way. It will even remember how to ward off foreign pathogens, foreign antigens, over time. Once the adaptive response has developed the first time in what's called a primary response, memory cells remain. Upon a repeated exposure, the response will develop faster and will be more robust. Note the difference with the innate response, which recognizes common molecular motifs and is always the same. This is a graph illustrating the difference between primary and secondary immune responses. The two arms of adaptive response. Adaptive immunity develops during an individual's lifetime. Humoral immunity involves antibodies, while cell-mediated immunity involves T cells. In humoral immunity, B cells recognize antigens by antibodies on their surfaces. The B cells become antibody-producing plasma cells. In cell-mediated immunity, the T cells recognize antigens by TCRs or T cell receptors on their surfaces. The terminology of antibodies. Serology is the study of reactions between antibodies and antigens. Antiserum. An antiserum is the generic term for a serum because it contains an antibody. Globulins are serum proteins. Immunoglobulins are antibodies. Gamma globulins are serum fractions containing antibodies. Here we see a generic view of antibody structure. In a generic sense, antibody structure is made up of two heavy and two light chains. Each has a variable and a constant region. In humans, there are 5 types of antibodies listed here. This is the IgG antibody. It is a monomer. 80% of it is serum. It has a fixed complement, can be found in blood, lymph, and intestines. It can cross the placenta, enhance phagocytosis, neutralize toxins and viruses, and protect the fetus and newborn. It has a half-life of 23 days. This is IgM, which is a pentamer. 5 to 10% of our serum is made up of this type of antibody, has a fixed complement, can be found in blood, lymph, and on B cells, and agglutinates microbes, and is the first antibody produced in response to infection with a half-life of 5 days. This is IgA, which is a dimer, making up 10 to 15% of serum antibodies. It's found in secretions, provides mucosal protection, and has a half-life of 6 days. This is IgD, which is a monomer, making up less than 1% of serum. It's found in blood, lymph, and on B cells. On B cells, it can initiate the immune response and behave as a B cell receptor. It has a half-life of 3 days. This is IgE, which is also a monomer. It's found in less than 1% of serum antibodies. It's found in mast cells, basophils, and in the blood. IgE is common in allergic reactions, and the lysis of parasitic worms has a half-life of 2 days. Here's a table summarizing the aforementioned antibodies. Antigen-antibody interactions can be described through affinity and specificity. Affinity is the strength of the bond between antigens. Specificity is the antibody recognizing a specific epitope. This can affect agglutination in bacteria, opsonization, complement activation, antibody-dependent cellular toxicity, and neutralization of toxins and viruses. Agglutination is the cross-linking of pathogens by antibodies. It's most effective using IgM and forms large aggregates, and is illustrated here. Opsonization. In opsonization, the pathogen is coated with molecules that facilitate phagocytosis. The phagocytes bind to the antibodies and ingest the foreign pathogen. Complement activation uses the classical pathway, where antibodies binding activates the complement. Antibody-dependent cell-mediated cytotoxicity, or ADCC. If a pathogen is too large to be phagocytized, the antibody will bind to the pathogen, then natural killer cells, macrophage or eosinophils, will bind to the FC region. The cell will release cytotoxins such as perforins and granzyme. Neutralization is the binding of antibodies to prevent attachment of pathogens or toxins to host cells, and it's effective against viruses, toxins, and some forms of bacteria. Major histocompatibility complex molecules, or MHCs. MHC molecules found on the surface of all nucleated cells. Major histocompatibility 1 complex is found on all cells, while MHC II is found on macrophage, dendritic cells, and B cells. The role of the MHC complex is antigen presentation. Non-self antigens are processed and presented to T cells, MHC1 to effector T cells, and MHC2 to T cells initial activation. This is an illustration of MHC class 1 and class 2 bonding. Antigen-Presenting cells, or APCs. APCs include dendritic cells, macrophage, and B cells. They possess antigens and present them in the context of MHC antigens. For dendritic cells and macrophage, pathogen is phagocytized, digested, and epitopes are displayed together with MHC2 on the surface. Antigen presentation with MHC class 1 molecules. Presence of MHC1 signals that the cell is normal. Self-antigens are presented with MHC1. Virus-infected cells will present antigens from the pathogen, targeting the cell for destruction by natural killer cells. Cellular immunity. T lymphocytes are the driving force behind cellular immunity. They are produced in the red bone marrow but mature in the thymus, hence the name T lymphocyte. Thymic selection eliminates immature T cells that react against self. It's important that T cells are tolerant towards the self to prevent autoimmune disease. Central tolerance is self-reactive cells that are eliminated by apoptosis in the thymus, while peripheral tolerance involves regulatory T cells inhibiting any self-reactive T cell. This is a T cell receptor, which is similar to an antibody, has a variable and constant region. Here we see classes of T cells. Take a moment to pause the video and review each class type, its activation and function. Helper T cells, CD4 cells, can be activated only by APCs or antigen-presenting cells, presenting foreign epitopes with MHC class II. Once activated, they produce mediators that orchestrate the immune response. Subtypes include T helper 1, T helper 2, and T helper 17. Some of these cells may also form memory T cells. This is a summary of the subtypes of T helper cells. Pause the video and make sure you're familiar with each before moving on. Cytotoxic T cells, or CD8 cells, are activated by antigens presented by APCs, but with the MHC I antigen. These become effector cytotoxic cells that kill directly. Cytokines released by helper T cells are important. They release perforin and granulozymes and cause apoptosis. Superantigens and unregulated activation of T cells. Superantigens can provoke an uncontrolled T cell response. Toxins from streptococci and staphylococci are examples of things that can lead to an uncontrolled T cell response. Cytokine storm and systemic inflammatory responses can occur. B cells are formed and mature in the red bone marrow. After negative selection of self-reactive B cells, the final stages of maturation occur in the spleen. Naive B cells are those that are not yet active. This is an illustration of a B cell receptor, or BCR. These receptors are membrane-bound IgD and IgMs. They can recognize antigens without T cell help, but it is less optimal. They are different from T cells in that they do not present with MHC, and they can recognize non-protein antigens. T-dependent antigens are mostly repetitive structures, short-lived, and have no memory and no secondary response. They are illustrated here. T-cell dependent activation. In T-cell dependent activation, a B cell recognizes and internalizes the antigen. It then presents it to the helper T cell. The T cell becomes activated, secretes cytokines. The B cell is activated. The B cell proliferates and differentiates to plasma cells. The antibodies are produced, first IgM, then IgG, and memory B cells are also produced. This is an illustration of that process in a graphic form. Classifications of adaptive immunity. There is naturally acquired active immunity, which results from infection. Naturally acquired passive immunity, which results from transplacental or via colostrum. Artificially acquired active immunity, which involves vaccination, and artificially acquired passive immunity, which involves the injection of antibodies. Herd immunity. Immunity or susceptibility depend on the individual's ability to mount an effective immune response. Herd immunity, if few individuals in the population are susceptible, that limits the spread of the infection. Herd immunity can be gained through exposure or through vaccinations. Variolation. Historical observation is those who survived smallpox became immune. Variolation is the deliberate inoculation of smallpox material into a patient, causing a weaker infection. This led to the modern vaccine and Jenner using cowpox. This is a table detailing the different classes of vaccines. Pause the video to briefly review each type of vaccine, the advantages and disadvantages, and organisms that they are used against. This concludes our coverage of Chapter 18, Adaptive Specific Host Defenses.